Industrial fermentation is the intentional use of fermentation by microorganisms such as bacteria and fungi, as well as eukaryotic cells and insect cells, to make products useful to humans. Fermentation at an industrial level is carried out in specifically designed vessels called fermenters or bioreactors. These vessels are utilized to support the growth conditions of microorganisms carrying out fermentation. Its design and construction must provide the optimum environment conditions to the microorganisms. Fermentation is effectively applied in many sectors, which include production of bioethanol for biofuels, sewage treatment, and the food industry. One of the fermenters currently being used is the stirred tank bioreactor. Let's take a look at how it is set up and operated. Parts. The fermenter consists of multiple parts. First, we have the vessel, which is made of borosilicate glass. Inside the vessel, we have the airtight seal, sparger, the foaming paddle, and baffle, not to mention the pH, DO, level, and temperature electrode to measure the magnitude of their respective quantities. On top, we have the motor that powers the movement of the vessel. On the outside of the tank, we have the sample bottle and the compressor that provides the fermenter with the air needed. We also have the heating plate at the bottom of the fermenter, which provides the warmth for the process, and the cooling jacket, which provides the necessary cooling element to control the temperature. Other parts include silicone tubes, supplement bottles, and general electronics. Calibration. Note: calibration will only take place for DO and pH. All other parameters have been pre-calibrated. The calibration of pH and DO has a 2-point gauging system with 2 standards that need to be calibrated. On the control unit, we have the pH and DO sockets as shown. Let's start with the pH calibration. Firstly, remove the pH electrode and connect it to the pH-based control unit labeled pH. Ensure it is screwed in correctly. Then, remove the tip on the electrode. Secondly, prepare the needed buffers and rinse the probe with the ionized water. Then, dry the probe with blotting paper. Thirdly, open the calibration page by clicking the setting page, followed by the calibration of the respective fermenter. Bear in mind that each fermenter must be calibrated, F1 and F2. Proceed to key in the pH of the buffer into standard 1. Lastly, dip the pH probe into the buffer solution and gently stir it until the pH value becomes stable. Click the OK button below the pH value. When you click OK, a pop-up window with a username and password should appear. Choose the Calib username and key in 1 as the password. Then click OK once more. Repeat the same steps for the second buffer standard. Next is the DO calibration, which is a bit simpler. The calibration page of DO is from 100% to 0%. Firstly, remove the DO electrode from its shell and make sure it is screwed in correctly into the base control unit labeled DO. Then remove the electrode tip and place it in a dry area Then proceed to the calibration page and click OK on the 100% standard. To calibrate 0% DO, simply place the DO electrode in any carbonated drink and click OK on the 0% standard. Sterilization. Step 1. Turn off the power to the controller and remove the motor from the top of the vessel and carefully put it aside. Step 2. Disconnect the air and all gas lines from the inlet filter on the sparger and disconnect the water lines. Step 3. Clamp off the harvest tube, the sample tube, and all other penetrations that are immersed in the media. Step 4. Disconnect all probes and sensors and remove their cables. If you are using pH and DO probes, install each probe shorting cap provided in the probe kit. Step 5. Wrap all filters with aluminium foil to protect from steam. Attach a piece of tubing wrapped with some non-absorbent material such as wool or cotton to each of the addition ports. Wrap foil around the end of tubing. Remember, before placing the vessel into the autoclave, to loosen the glass sampling tube by half a turn to alleviate excess pressure. Fluid flow. The two fluids in question are air and water. Air acts as a sparging element, while water is mainly used for air removal and in cooling control. The water source enters from the first hole from your left. It then partitions and leaves number 2 heading to the condenser and hole number 3 heading towards the water jacket. The water entering the condenser cools the gas and exits towards the drain, followed by the water leaving the water jacket. The water heading towards the water jacket is controlled by a solenoid valve that will open upon the temperature rising above its set value or range. Before we can do a run, ensure that the valve inlet is connected to the unit. Make sure the drain line is properly connected to the exit of the condenser and also the exit of the water jacket. Connect the water lines to the vessel jacket and exhaust condenser. Once everything is connected, turn on the system and let it run and inspect for any leakages. Supplement and sampling. In order to maintain parameters, we might need to add additional material to the fermenter, such as to control the pH or the foaming inside the vessel. In order to supply these contents, a peristaltic pump is used. The peristaltic pump squeezes the contents through the rotation of tubes from the feeding bottle to the fermenter. When setting up the peristaltic pump, ensure the supplement bottle is equipped with a filter as to ensure fluid flow and protect from inner and outer contamination. Then connect a silicone tube from the feeding bottle into the feeding pot on the top of the fermenter. Lastly, on the halfway point of the tube, place it into the peristaltic pump. Press the manual on the button to secure the tube around the pump, and lastly, push it into place and close the lid. Repeat each step for anti-foam, feeding, and base. Lastly, the sampling. Before the sampling, ensure that all tubes are clamped. The air sampling tube is closed accordingly, and all tubes have been connected and fitted with filters. Firstly, connect the syringe to the syringe tube. Nextly, unclamp the harvest tube and pull the syringe. This will allow the liquid from the vessel to flow to the sampling tube. After that, clamp the harvest tube and unclamp the collection tube. Then push the syringe and watch the culture flow to the collection tube. Unclamp the air sampling tube and clamp all other remaining tubes. Repeat steps until you get the number of samples required. Inoculation. Now that the vessel is sterilized and running, there's one more step before we can continue the fermentation. Inoculation is the addition of microbials to your culture to sterilize and overcome any lag in growth due to the possible contaminations of bacteria to the surroundings and vice versa. A firing, basically a jolt of flame from a Bunsen burner, should be placed near the inoculation port. Ensure when opening the inoculation port that it is only turned from the top and no contact is made with the lowest part. Next, place the inoculation ring near the port and pour the inoculant in the presence of a flame. When all the inoculant has been poured, run a flame under the inoculation pot and close it firmly. The inoculation must strictly be poured in only after the sterilization and fermenter setup.